Hi everyone, I am Russ Connelly. I am doing my PhD in this field of fantastic finding needles in haystacks. We are trying to find seahorses independently of the vision of trying to actually see them that way. And so I'd like to thank all of the collaborators, funders, and everybody for that, because without those people to help, we wouldn't have this cool project. So we've got two seahorse species in the UK, Hippocampus hippocampus and Hippocampus gutulatus, both data deficient on the ICN red list. They're also CITES listed, but pertinently for the UK, they're listed under the Wildlife and Countryside Act of 1981. But that essentially means citizen science is null and void. We cannot go out and try and find seahorses on a citizen science approach without having to use a permit. And so it's actually an offense to go out and try and find seahorses in the UK without actually having that permit. So it's quite difficult. So we're using their biology against them. They are sygnathids, as Nuno said, which means they're together in fused jaw. And because of that, we hear a distinctive clicking sound. And that is distinctive to the species that we're looking at here. They're voracious predators, again, so we're using their biology. They constantly need to be feeding, so we're trying to hear those clicks each time. They are very poor swimmers and have small home ranges as well, so that we are able to find the seahorses much easier in a small area. And then they're also that umbrella species or flagship species, as Nuno mentioned, because we can protect the areas that they're surrounded in to then give everything else a good chance to survive. So they are the celebrity fish, and in the UK, because there's this mystique around them, you can't actually be able to find them, as I said. All we can do is look on Facebook, and we can search seahorse found or seahorse sighted. And there are just incredible amounts of people being like, wow, we found a seahorse, that's amazing. And so it hits the news fairly often. That one's my favorite, because we just found that in the airport. So this is the way that we're currently detecting seahorses using the Natural England dive team. So Natural England are the people that are allowed to go out and survey through the government funding, and they go in an area and they go in a U-shaped transect search. So they've got two divers here in a buddy team, spread 2 meters away from each other, go along a transect and try and find the seahorses. Obviously, this is all visual, and it's high costs because of health and safety requirements in the UK. You have to have an additional course to be able to actually go out and try and find them. And all of that racks up cost. It increases time, it increases human effort. It can't be done on any sort of shallow environments just because everything has to be done off a boat. And it can't be done in turbid waters. And as you can see, that was one day after another. Literally, the first day I found 4 Hippocampus gutulatus on the left within an hour. The second day, you should be able to see my hand in that image. It just isn't possible. So we need to find ways of visually, independently trying to find seahorses this way. So I have 2 methods in my PhD. It's using passive acoustic monitoring and environmental DNA. I'll be talking to you about the environmental DNA section in my poster presentation later on. But we'll be talking about the passive acoustic monitoring for this. You can break that down into theory, testing everything out in aquaria, and then moving into the in situ fieldwork side of things. So, passive acoustic monitoring. It's essentially listening to the ambient sounds that are in the area. And what we do is we've got these things called hydromoths. All we do is we place those down and record whatever we hear. What we can do from that is collect all the data, corroborate it with the GoPro here that we have, and listen to them. These seahorses start making feeding sounds whenever they feed, and we can apply some band filters to remove the unwanted noise, such as the pumps or any sort of filter noise and background from the intended signal. You can save that, annotate it, and then progress it down the line. This is hopefully going to work, but hopefully you should be able to see and hear a visual confirmation of a seahorse feeding click. So there should have been 3 clicks that you could be able to hear there. On the right, we have visual representations of the seahorse feeding clicks in sonograms or spectrograms. And what we can do is result that into the field. And as you can see here, the visibility is absolutely poor as well. So what we place the hydrophones down on the sandy sediment here using land anchors. Obviously, I couldn't bring the land anchor in just because I traveled on hand luggage, and that was a bit of a stab risk. But what we can do from that is place some band filters around. It's 0.1 to 3 kilohertz in range, which is where the main energy of where the seahorses feed. We record for 24 hours and retrieve it after multiple days, and we find out what we've got. This then leads me onto the deep learning side of things. So what I did was collated, I think it was 1,300 different feeding clicks from seahorses, trained a convolutional neural network to then be able to confidently identify the seahorses feeding. So it then is broken into smaller, easy-to-view chunks in around 2-second intervals, and then that trains it, validates it, and gets it accurate in that sense. So this is the result from the convolutional neural network. As we can see here on the MEL spectrogram, this is an output from the neural network. It is 100% confident that that is indeed a hippocampus scutulatus, and I verified that later on using the GoPro footage that corroborated it. On the right here, you can see the confusion matrix, which is how well the model performs. So it has 0 false positives, which is a great result, because then we're not saying that there was a seahorse when there wasn't, which is the whole point of what we're trying to find out. There are slight false negatives, but that can be overcome with just time listening for the seahorses. On the further down, you can see how well the model trains and then gets validated across epochs, which is the time taken to generate that model. We can then, there might have been, that are feeding clicks at 1 meter, 5 meters, 10 meters, and then progressively it gets then in towards more of this, towards 60 meters, which we're getting some sort of background noise in that sense here. This is done in Studland Bay, and you can see here that's how far away we could detect a seahorse. So it's now up to 60 meters away that we're able to do that. We need to make sure in a statistical manner that we are detecting seahorses. So this is where machine learning comes in, and this is the random forest model. So what we do is we generate the pipeline of clicks. We've got cygnathids here and 2 different pipefish species, and then also a snipefish just for an outgroup that does also make sound as well. We can use that random forest to generate an understanding of which species is what to that level. And we have a 5-fold cross accuracy of 92%. So we are 92% certain that whatever we put into it comes out with one of these 5 species. So we can put this all together. And what we do is we have the acoustics, we place that down in situ where we're trying to find a seahorse, where the turbidity is not great, or there are restrictions on whether we can actually dive there, or anything like that. It generates a wave file. That can then be put into a supercomputer at the university that we have, which is trained and validated as the convolutional neural network. It then outputs what we think is a seahorse. We need to then make statistical models, which is the random forest model, to then be able to determine whether we have found that seahorse. And we found it. Yeah, so the previous audio that you're hearing were from Hippocampus scutulatus in the seagrass areas. Thank you very much. That was a quick, brief one. I also want to plug, I'm finishing my PhD in about a year, so if you want to collaborate and do anything further, please get in touch.
Well, thank you. You have minutes left if you want to ask for questions. Somebody want to? Oh. 3 minutes left, okay?
Yes.
Were you able to distinguish those species
We are, yes, yeah.
What different frequencies?
Yeah, so it's different frequency and different timings of the seahorse clucking events, yeah. So hippocampus is a shorter event than gutulatus. So we were managing to work out, I think it was 18 different parameters for differentiation between sygnathids.
And the clickings are all only associated with feeding? Or?
Yes, yeah, so there are other noises that are created, such as growling if they're grabbed or anything like that, but we were only focusing on the feeding because we can't even touch them with our permits, so it's just not possible.
Yeah, okay. Very, very interesting. Thank you.
Thank you, Russell, for your presentation. That was my question also, if you saw any differences between the frequencies, the kilohertz, because we are talking about kilohertz, right?
Yeah.
It's really high. If you saw differences in the species of this range of frequency, did you? What is the difference between the two species? I did not got it.
Yeah. So essentially, it is the length of the snout that impacts the frequencies of the sygnathids. So the longer the snout, the higher pitch it will be. So a higher frequency. So for the snipefish, which are continental wide, is so high in comparison. It's now more like a snapping shrimp in sound.
Hello. So I think that was a really cool idea. So I just want to know, like, how did you find this? Like, how did you come up with this idea that you could look at the sound?
Yeah, so we knew that seahorses and sygnathids are soniferous species when they feed. I was approached in this project for my PhD just to work out whether we can detect them in situ without having to worry about any sort of the regulations and that side of things, because in the UK it's just so difficult to be able to progress any sort of science on that sense.
Thank you so much.
I wonder if you saw any differences in the different type of prey. Do they sound exactly the same when they're eating different things?
Yeah, so we were looking at mysis and also artemia, and both of them were the same sound from that.
If you're relating the size of the snout with the sound, is there an overlap when they grow between one and the other species where you can do some...
Yes, for sure. So when they start growing, hippocampus becomes more like gutulatus originally, but that's something we need to look into further for sure.
Thank you for the presentation. You say that you were able to distinguish the species, and what about the individual?
Not to individual level, I don't think. I think potentially with future technologies, but this is just not there yet, no.
Yeah. Is your code open? Can we replicate it for other species?
There should be, yeah. So once I finish and publish, it should be available for everybody.
Okay.
Thank you very much.